All right, welcome back for the seventh lecture of 7 on the course Biodiversity and Biotechnology and Biorefineries. And in this seventh lecture, we're going to finish the subject conversion step in biorefinery. So what does a catalyst do? I should add that next time. So what does a catalyst do? The working principle of a catalyst is that it enables a faster chemical reaction. So turn up volume. Okay, so welcome back. I just boosted my microphone levels up a bit, so maybe you can hear me better. The working principle of a catalyst is that you quicken the chemical reaction. You make the chemical reaction go faster by decreasing the activation energy. This destroys compounds and builds new compounds, but the catalyst is not consumed in theory. However, anybody who's worked in a factory knows that catalysts action. We're unsure exactly why it happens, but in theory it is not consumed. And there are multiple chemical transformations. So what is activation energy? Activation energy is the definition is the energy required to raise a molecule to its transition state to undergo a chemical reaction. So energy that must be overcome in order for a chemical reaction to occur. This means is there's a activation energy needed for a reaction to happen. It's like if you are in a standstill and you want to move forward, you need inertia is keeping you back. Right? An object in motion tends to stay in motion, and similar to that, a molecule that has a tendency to stay in the same form. So you need a little bit of energy to push the reaction forward. An enzyme, a catalyst, lower the activation energy. An example of this is an enzyme here you see. Here's the substrate. Enzyme locks into the substrate, and that lowers the reaction energy of the reaction. But I think this is all a review for everybody. Are there any questions? Well, I think everybody you learned about catalysts. Well, that's a good thing. That makes, a good starting point. But the different types of catalysts. So we know the definition, and we just had a short review. But then what are the different types of catalysts? You have biocatalysts, which plants, animals, insects, but also enzymes, yeasts, algae, and fungi. They are enzymatic, organic, and inorganic catalysts. You have chemical catalysts, which are catalyzing compounds, including nickel, copper, medium acids, and bases, including platinum, nickel, iron, we saw in the previous one for pyrolysis. And you have biological catalysts. So again, I'll get to that. Thank you, Leonard. I'll get to that. Enzymes, electrochemically active bacteria, archaea. Well, plants are very interesting sites for fungi to grow. So, if maybe you've heard of fuel cells, plant fuel cells. So if you have a grass, what they do are organisms that change the potential energy in the soil. So they reduce iron, for example. And this reduction creates energy. So while the plant itself is not reducing the iron, it's in a symbiotic relationship with microorganisms that do. And plants are not the only organisms that do that. There are some animals in the ocean that have that. They have bacteria in their blood for example. It's like an octopus that has that. It's like will glow in the dark. So what it's doing is it's actually reducing. They have bacteria in their blood that reduces, uh, or in their skin that reduces the iron and produces light energy. So plants and animals can do it, although we don't use them in industrial. Can you imagine, like a tank filled of octopuses making energy? It's like what's it called? Steampunk fan. Potential is very, very low. However, for plants, grass, it does work. I have colleagues in Wageningen who are doing research on the use of grasses to create energy. The problem is it does consume the plant. Plant will die. You just have to, I don't see that as a big problem. You just sow new grasses, and the grass could be used. Right, steampunked octopi. Yeah, I want to cut the cut of that, Shores, if you get any money off of it. But yeah, yeah, not a lot of industrial applications, but they do it. They do it. Chemical catalyst, so base as a catalyst. So a base capable of deprotonating an alcohol. Its overall chemical reaction requirements are methanol, base, and 0 water, so you need almost no water. Here you have a type methanol, which makes glycerol and methyl esters. This will happen under room temperature, and it's a good way of making glycerol and methyl esters from. Another chemical catalysts, they create simple chemical reactions. Disadvantages are that they have chemicals as waste, and the recovery is energy intensive. So you can reuse them over and over and over, but like as I mentioned yesterday in using ethanol, getting ethanol, distilling ethanol from ethanol water mixtures costs a lot of energy. So if you want to, here you see it here. Well, chemicals as waste means that you use a base, you use a chemical reaction, but here it produces things you don't necessarily want. You may not want glycerol. And then how do you get the glycerol out? Well, you get it out eventually. Here's the 90% crude glycerol. And you can recycle the methanol, but you have to get it out somehow. Right? Because that's one of the things we're talking about as a catalyst shouldn't be destroyed, but in theory. But unfortunately, in practice, it is not destroyed, but it's wasted. You have to keep on, for example, steam explosion. Steam explosion, you just need water, heat, and pressure. Here you need to buy chemicals for it. Now we can move on to enzymes, yeasts, bacteria, algae, and fungi. There are a few techniques that microbes, fungi, etc., which include fermentation is a very widely used one. Fermentation, it is catalyzed by enzymes. In the video this morning, we showed one of them, yeah? So you had a bacteria which created an enzyme, treated it. It can also be catalyzed by the microorganisms, cells, so microbes or cells of yeast, bacteria, and algae. Enzyme fermentation can be represented here. So you have an organic feed plus A. With the enzyme as catalyst, it has a product chemical R. So A is transformed with enzyme E to produce R. Non-enzyme fermentations or direct fermentations from the microbes. You have, again, here you have organic the microbe C, which gives you your product R plus more cells. So they use the feed for splitting their cells as well. So they ferment just like bread, right? So bread or cheese or beer, the microorganisms transform the sugars into alcohols, which is an organic solvent. And they also produce more microorganisms, which is good because they don't live forever. Enzymes, it's a large biological molecule responsible for thousands of chemical reactions. These include proteins, so amino acids, and they have an active site on the enzyme, represented here in the figure. The overall chemical reaction will be some kind of sugar plus water, and the N just signifies a certain mole ratio. Don't get too hung up with the N. That just means there's like 2 of these and 3 of these. It depends on what exact sugar you have here. And it's transformed into a new product as it consumes the water. It requires a solvent, and in this case we're using water. You can also use an ionic liquid or an organic solvent. Probably never heard of ionic liquids before. Ionic liquid is basically a liquid salt. Yeah, so it's a positive, negative collection of molecules in liquid form. You're used to seeing salt in solid form, right? Every day I put salt on my on my food, but you can also have it as liquid form. And organic solvents are ethanol, methanol, isopropanol, etc. Advantage of enzymes is highly selective in superproduction. That means you can put it on a substrate, a certain location. You don't have to necessarily mix it. Disadvantage is a limited lifetime, so it's not, even though it says we say it's not consumed, it actually is consumed. Hampered by inhibitors, so if there's anything that's in the way, like lipids, etc., it stops the process. There will be ex situ production that you don't want. So you want to produce something with an enzyme in a certain place it's like a molecular cage. But it'll also be in other parts. Remember, an enzyme just speeds up the process. It'll take place in other places without the enzyme or the catalyst. It also requires a sterilization step to make sure you have pure. The costs can range greatly. Some enzymes are very cheap and some enzymes are extremely expensive. And then, I mentioned this maybe a few days ago, you get them back. Enzymes are teeny tiny, dissolved in your solvent. How do you get it back and use it again? Almost impossible. Well, it's possible, but it's very expensive. You need to use probably chromatography. Microbial fermentation is, here it says here, a hodgepodge of foods and cells, bugs in the layman's term, which are all reacting. So in microbial fermentation, it's not one specific strain or necessarily species even of organism. Even the genuses can be and it's represented here, and the A with T plus residue. Very interesting, often use of microbial fermentation is the breakdown of water treatment, producing cells, like yeast growth, and the cell's waste material, alcohol, and antibiotic production. And there are lots of other things, not just antibiotics, but insulin. You can modify a yeast cell or lots of cells to produce whatever you want. Itaconic acid, but especially things like insulin, all produced. It used to be isolated. People who needed insulin injections, insulin used to be very, very expensive because they would just isolated from blood, from human blood. Nowadays, it's extracted by a microorganism in basically a petri dish. It's much, much less expensive. Biocatalysts, such as yeasts, they are eukaryotic. They have a cell nucleus. They are uni, but also multicellular chemical autotrophs. So they use chemicals rather than synthesis. Yeah, eukaryotic. Means with in Greek, and karyote means seed. So they have a nucleus. That's a good bonus point for people who are taking cell biology, which is not everybody, but I think a significant number of you are taking cell biology. I think now it's great because, yeah, nucleus. Yeah, nucleus is you have a cell, and the cell is filled with a liquid, and within the liquid there's another area called the nucleus. So if you don't know what nucleus is, just Google. Yeah. Nucleus is correct, Christian. But literally it means with seed. Ah. The eukaryote just means it has a nucleus. It doesn't have free-floating DNA. It has DNA in a nucleus, among other things, not just. I'll continue. Yeah. Requirements is solvent, mostly water, glucose, right, because it needs to get its energy from somewhere. Oxygen as the electron acceptor. Of course, carbon and nutrients. This is all the things that you're learning in cell biology, I think. Might have gotten also microbiology. Conditions, it depends on the yeast, but most yeast don't like anything below freezing actually, but -10 to about up to around 40. And because this is, we have to do with electron transfers, pH should be slightly acidic. Just a little bit. The advantages of yeasts are they have high selectivity, and they are in situ reproducible, right? So here you have a reactor. It's just a big glass bottle, and it will break down sugar and make whatever you want. So ethanol, for example, glycerol, or acetonic acid. So it's very selective, and it's everywhere in here. However, disadvantage is it's hampered by inhibitors. It doesn't like what doesn't like what it doesn't like. If it can't use it, it hurts it, including that thing which you are making, right? So when we make beer and wine, we ferment it and produces the ethanol which we want, but it's inhibited by the ethanol. Yeah, so at a certain point, around 12%, yeast stop working at 12% ethanol because it's inhibited by its own product, which you are extremely cheap. Yeasts are very cheap, and they can be reused cells because they have a life expectancy. However, they produce quite quickly. Every 20 minutes, they split. Produce. The biocatalyst mixed culture bacteria, so you can be prokaryote organisms, and I can hear Christian saying, well, what's prokaryote? A pro meaning before the seed, so they do not have a nucleus. They just have a cytoplasm, but no nucleus. So they have free-floating DNA, RNA, organelles. It's all just one big mixture. There's no nucleus within the cell. Requirements, again, solvent, E acceptor, E donor. In the previous case, that was sugar and oxygen. Again, carbon and nutrients. And here, the temperatures can go slightly higher. Yeah. and that's because it's a mixed culture. So have one type. Advantage is a mixture of bio-waste, so you don't have to have one type. And it's in situ. You don't need to sterilize it because it's its own habitat, right? So it's self-sustaining. Disadvantage is microbial competition. So you have a mixture, and some will perform better than others. They're very cheap to produce and can be reusable many times. So here are things that you can make from fermentation. These include C6 sugars with yeast ethanol, starch, amylase produces glucose, ethanol, cellulose, glucose, guess what? Yeast ethanol, hemicellulose, C sugars, yeast ethanol. You can, all of these pathways, yeah, it all produces ethanol here, beer, wine, most of us enjoy that. Types of fermentation. Yeah, it just depends on what your starting fuel is. Yeah, so this all goes back. We're rounding this course off. It's going back to some of the very first parts of the lecture, of the series. So here you have, if you have a C6 sugar, glucose, hey, that's easy. Just produce ethanol. The sugar is available for the yeast to use. If you have starch, right, which is mostly amylose, then you need to use what we talked about, an enzyme, amylase, to break it down into glucose. And again, the yeast can change it to ethanol. Cellulose, right? You could use an enzyme like cellulase, but you could also use an acid treatment to produce glucose and use the yeast, the ethanol. Here's the hemicellulose. Here you can use enzymes, but also just this morning we were talking about that. So here I'm using, I say enzymes, but you can use all sorts of physical processes. Don't limit yourself to just conversion technologies. You can also use the pretreatments. And if you use the correct pretreatment for the correct biomass, you don't have to do certain steps, conversion steps. You have to think about that. So in your projects, you may have lemna, which has no lignin, which is extremely helpful if you want to get to the sugars. So extract the proteins first. Now you're left with cellulose. Well, maybe you can acid treat this cellulose to make glucose. And then the glucose, you can make ethanol. So now you're not just making proteins, you're also producing ethanol. So you have more than one product. You have proteins and ethanol. So you have to be clever, guys. Come up with your own biorefinery concept. Is wood. Now we're doing both pretreatment and conversion. So wood, which is cellulose and hemicellulose and lignin, you can pre-treat it, go through saccharification. Saccharification means a conversion to sugar. So that's exactly, this is all saccharification. A saccharin is just sugar. Saccharification to make the starch or the cellulose enzymatic or whatever. Sugar beet already is very sugary. So now you can ferment to make ethanol, for example. And through distillation, you have bioethanol, right? So the only difference between, I should ask this question, I'm just going to tell you. The only difference between bioethanol and ethanol is that bioethanol is made through fermentation from a factory. Other than that, it's exactly the same as normal ethanol, which you can get from other. This is the end, guys. It's the last. That must be a good feeling. This is the last 25-minute lecture. Are there any questions before I cut off? The lecture and let you do the assignments. I don't see any questions. Then, oh, well, this is like a quick question, Christian. I will get to that. Let me just pause the, okay. So I've started the recording again, just so people know what the question was that Christian posed. Christian asked me to explain a research question, and the research question for assignment 7 is to search the internet for a biorefinery type that your product group has been given. So you actually, this year you all have a green biorefinery. Yeah? So there's slightly different biomasses, grasses versus sugar beet leaves versus lemna versus macroalgae. But use the internet and find a biorefinery about that. Like, for example, Grasa, it would be an example. The person who gave you a presentation yesterday afternoon, and then say, how could this be used in the gasification process? So here you have, let's say, for example, I'm trying to think of an example that you guys are not doing. So you have a green biorefinery. Let's say press cake. You are assigned press cake from or coffee beans, right? So the Starbucks has big factories, and they make pre-made lattes you buy in a grocery store. Yeah. So in this factory, they use coffee beans, and they're left over with coffee grinds. And even though I'm not completely familiar with coffee grinds, I imagine that they are very high in lignin fibers, right? Fibers, but also it has some cellulose, hemicellulose in it. The question is, how could I use this leftover waste in the gasification process? At what temperatures would I have to make it? Where would it come from? So I'm saying Starbucks, but it could be any coffee place. Temperatures, how would draw maybe a diagram, balances. The same thing for pyrolysis. At what temperatures would you shoot the pyrolysis? So go back, go back to these sheets, maybe even the sheets that were earlier. Where was that? Yeah, so here's pyrolysis. But so recreate, please don't copy paste this work. Just, make your own diagrams. So here's sort of biomass. I would write coffee grinds. Pyrolysis with no O2, and condensation of liquids. What liquids? What gases are produced? Maybe use the reaction. For gasification, use reaction like this. The first two are actually really easy. The second one, the third one, you can talk about Fischer-Tropsch synthesis. So which metal would you use? Would you use cobalt? Or would you prefer to use iron? So cobalt or iron? So it doesn't take you too long. I just want you to go back and add your own ideas. So make a few boxes, label a few things. Instead of just biomass, you write lemna, or you write, and before that you say, first we extract. Should we start with lemna? Then we use a mill to extract the water. It's left over, which is the fibers, etc. And you do that again for gasification, and you do it also for Fischer-Tropsch. Here. biorefinery design. Completely correct. So what basically, I'm trying to bridge, because you know when you, everybody's the same, I'm the same way, and I take a course, and the first part is just basically tell me what I told you, and the second part, the research question, is now take what I told you and add it and apply it to your research. So this one, I want you to think, okay, we have lemna, we have grass. Okay, first we're going to pre-treat it to get out the proteins, which are in the liquid phase, with leftovers. What are we going to do with it? Are you going to use Fischer tropes? And I'm saying now, show me how that would work, the Fischer tropes and the gasification and pyrolysis. It's like a downstream. Did I answer that question? Yeah? Okay. Okay. Well, I'm going to stop recording, but I'll be here for a little bit.